try to give you a little bit of an overview of a flavor of what we do at UCVM on pathogen genomics. We focus this time on parasites, worms. You have all other kinds of parasites, but I only look at worms, bacteria and viruses. And we talk about nema, biome, microbiome, and virome. Anything with the word "ome" on it, that's what we look at. And we try to analyze the diversity of these omes in various practical circumstances. So you're not only looking in laboratories. If you look at your own body, as you sit here, the amount of bacteria on and in your body will outcompete your own cells by 10 to 1. So basically, if you be a bit less self-centered, we are basically bodies carrying around humongous amount of bacteria, viruses, and maybe parasites. We are not so interested in rumen microbiomes. A lot of people working on nutrition have looked at it, and they came up with over 7,000 different bacterial species in the rumen. We're not looking at it. We look at respiratory tracts, especially in BRD-related issues. And our first studies reveal a humongous diversity of bacterial species, and I'll give you a flavor of that. And we want to understand what these commensals, so the bacteria, viruses, and parasites that actually live in and on you, play a role in your health, of course. And we use several approaches. For bacteria, we use ribosomal RNA. If you don't know what it is, it's a part of your protein-producing machinery. It's very conserved, very old-fashioned DNA, very similar in almost every bacteria, and that's why you can use it to identify most of the bacteria. Not all, but most of them. We know much about cultured bacteria, but what about bacteria that doesn't look like a bacteria, that has a little bit of a different appearance, or are very rare, but maybe still very important, or they grow super slow and you're not able to culture because you don't wait long enough. And sometimes, and most of the times, they're not even culturable. So we will place also some attention to culture negative outcomes. In my field, where I'm normally working, I'm a virologist. My official title is Global Health and Infectious Diseases, but my viruses are my hobby, basically, where I waste most of my time on. I use a kind of standardized PCR to identify different viral families. They are super diverse. We don't have a gene that we can target. And we break down DNA and RNA as much as we can because these viruses are very sturdy, and we want to get rid of host and bacterial genomes. And with that, we sequence everything that remains, and then most of it is still not useful. Dr. John Gilliard's lab is looking at antibiotic drug resistance, and he's doing research on different aspects of that. And I will show you a little bit of an idea of what you can expect out of analysis that he's doing currently out of populations of nematodes that he finds in different parts of the world, especially North America. He's using C. elegans, but Haemonchus contortus, a sheep worm, is his pet worm, but he looks at other ones as well. So basically, the study of the nema biome is following a route that you can extrapolate also to bacteria and viruses by finding your sample of interest and prepare the DNA, find your piece of DNA that you're interested in and amplify that with PCR, make a lot of that same product, and with that product you can sequence it in a new Illumina next-gen sequencer, basically sequencing anything that comes in that's a sort of DNA. And that has its challenges. The bioinformatics is then a huge chunk. It's tucked in the left corner. Basically, it's more than 50% of the work to make sense of 100 millions of reads sometimes. It's a specialized trait. So what you see here in Canada, and in special different animals over the provinces, you see that in the provinces more to the east, you see a different population. I will not go into many details because it's not my field, but you can see in the red bars that coparia is prevalent only in the western, in the eastern part of Canada, and in the western part hardly visible. So there's quite a fluctuation. If you compare that to what is known in literature, and if you say, well, what is known in North America, let's put it like that, it becomes a bit difficult to make that extrapolation. This is the situation in the Midwestern part of the United States. So it's not very difficult to see that the worm populations are completely different. It has no relation at all in what happens here in Canada. So if you want to make any sort of inferences on worm and helminth resistance development and influence on livestock production of your helminth population, you have to look here on the ground, and you cannot just say, well, they did a lot of good work in the US, therefore it's no use to do anything here. The microbiome study I was more involved with, basically what we've done is a very simple study. We mimicked the current situation in beef markets. So we brought animals to the market and brought them back somewhere else and see what happened in the bacterial populations in their nasopharyngeal areas. This is basically what we found. Before the market, there were steers about 500 pounds. You see a lot of green. Green is a mycoplasma. Mycoplasma in all kinds of varieties, so not only pathogenic mycoplasma, but they're all over the place. And the other part that's pretty prevalent is the Pastorella. Pastorella seems to be hanging out in these type of animals for long, and Moraxella here and there. But if we bring them to the market and swap them again, you see a lot of Ralstonia. Ralstonia is a bacteria we don't know much about, and we have doubts whether this is a correct answer. The reason why we have a lot of doubts is that we sequence every freaking piece of DNA. And even in our DNA prep kits, believe it or not, there is DNA in there. And now Ralstonia might be just a fluke in our preparations. And the reason why we are quite suspicious about it is if you see the before and the after market, they have different kits, or at least a different moment that they were prepared, and there is no Ralstonia in it at all. So that is causing some concern. But if we just remove that red graph and just focus on the before and after market visit, what you will see is that Pastorella has totally gone and are unidentified, but we have no clue what kind of bacteria take over, and mycoplasma. What it also means is that and this is the top 10 bacteria that we found, all the 90% other ones we left out. But we also understood is that if you look at the population of bacteria, and here you see the yellow, that is the aftermarket, the green is the just arrived back from the market, corrected for Ralstonia, and the blue is what we actually found before we brought them to the market. You see the bacterial population completely change, and they don't refer back to the old state. So doing something with a cow, bringing him to the market and putting him somewhere else will change the microbacterial flora 100%. Totally flips it upside down, and that has consequences. For example, BRD. So what we want to understand is what actually is going on in the microbiome of calves from birth to slaughterhouse. We want to know what happens if you give it an antibiotic. What happens if you give it a vaccine? And we have to understand what the host genome, okay, there we go, how that influences the microbiome. And I will finish with my own work. The BVD research, what I've done for the last 5 years. And basically, I focus on persistently infected animals. Persistently infected animals are animals that have been transient infected during life or came out of a persistently infected animal as a mother. And during their fetal stages, they were infected with BVD, and they got infected in such a way that they never get rid of the virus anymore. It becomes a self-antigen. That means there is no immune response. And for me, that is a very nice model to study what happens with a virus if there's no immune response messing things up for me. So that's why I use BVD as my favorite pet virus. And we see a lot of things happening. This is a picture out of 26 persistently infected animals we found on a beef farm in southern Alberta 2 years ago. And there was a beef producing enterprise without vaccination. And what you see, the whole yearling crop became persistently infected, and we got only 26 out of them, and they all ended up in my freezer. The prices were not that high at that moment. But, oh, can I go back? Yeah. So what you see here is that there are variants here that we cannot 100% explain. We have some idea what it might be, but why the heck is that virus changing so much while there is no pressure on it? What is actually driving these kind of changes? That's what we want to know. We understand now how viruses move from mother to fetus, how variable the virus actually is. That can help producing a vaccine that's more effective than that we currently have, because we know where to focus on now. And the other thing that we're now currently doing is to understand how viruses actually spread through Canada. And we started with a little bit of Western Canada, Saskatchewan, Alberta, and a couple of them in British Columbia. And what you see happening is that we now understand where viruses actually move and where are the big drivers of doing that. We can learn when viruses are introduced. So we have an idea when BVD actually was introduced in Western Canada based on sequences. That was about 1909. Maybe makes sense, maybe not. We're not 100% sure. Because we're not 100% sure, we want to make it bigger. So we now collected all BVD that is available somewhere in Canada in our freezers, and we do this again in a larger scale and see if 1909 still holds. What do we want to learn? In the microbiome study I just showed you from Edward Timsit, we now incorporate also the virome project, basically identifying all viruses that are present in the respiratory tract of calves. We want to know what their involvement is in BRD. And on the other side, with the BVD evolution, I would like to know how did BVD actually evolve over time as a pathogenic virus? How did it spread through Canada? And what are the influences of vaccines on BVD evolution? So it's nice that you vaccinate, but you might drive the virus in a certain direction. I would like to understand what it actually is. And what will happen next? How will BVD actually evolve? What will be the next variant that will circulate? We have no clue. We always are behind. We always wait until it happens in the field and then start producing a vaccine. But what about if we can produce a vaccine that's ready for a virus that will circulate 10 years from now? It's possible because the possibilities of these viruses are not indefinite. And we can identify all options of that virus, and why not make a vaccine for all options?